Ever since I was in middle school, I loved to look at the fins of our class's pet betta fish. His vivid colors were always lovely to watch, and he would flaunt his fins around constantly. But because of these vibrant colors, he can get a bit territorial when he sees another fish with a brighter pigment. But what causes a betta's fin color to be expressed? From the color of our eyes to the color of a betta fish, they are all expressed by what is known as genes. A gene is what determines what is expressed or what is not expressed in a characteristic of an individual. Genes are located on chromosomes. Within these genes are a set of alleles that express a characteristic. These alleles are usually represented as letters, known as the genotype. Each letter can either be dominant or recessive. Dominant alleles are indicated as the capital letters and are capable of masking over a smaller case letter known as a recessive allele. For example, we'll use a letter to represent the color of our eyes, or the phenotype, which is the physical trait that associates with the allele or gene, like this jagged leaf or this blue eye. Let's use the letter B. The capital B represents brown eyes, and the little b represents blue eyes. When an individual carries this pair of alleles, big B, little b, in their genes, the individual would be expressing brown eyes and not blue eyes. This is because the brown allele, capital B, is dominant and masks over the blue allele, little b, which is recessive. The recessive allele will always be masked whenever there is a dominant allele present. However, if there are two recessive alleles, little b, the individual would express blue eyes, since there is no dominant allele present. Whenever there are two of the same capital letters put together, or two lowercase letters put together, this is known as homozygous. You can remember this by remembering the prefix, homo, which means the same. The two capital letters are known to be homozygous dominant because, as said before, a capital letter always means it's dominant, and there's two of them, making it homozygous. The two lowercase letters are known to be homozygous recessive, since the smaller case letter is a recessive allele, and there are two of the same kind. When there are two different letters together, like a capital and lowercase b, this is known as heterozygous. The prefix hetero means different, just like there are two different types of letters. Now, using the genotypes for eye color, let's say that there was a father who was heterozygous for brown eyes, and a mother who was homozygous for blue eyes. What is the genotype for both these parents? Since the father is brown-eyed, we know that the allele must have a capital B in its genotype. But there are two possibilities for this brown-eyed individual. He can either be big B, little b, or big B, because both genotypes express brown eyes. But since the scenario stated that the father was heterozygous for brown eyes, that means he must be big B, little b, since big B is homozygous dominant for brown eyes. Looking at the mother's genotype, we can see that since she is blue-eyed, she doesn't have the capital B in her genotype that represents for brown eyes. So she must have both her letters as little b instead. It's good to know the genotype and phenotype of an individual since it leads to knowing how to set up a monohybrid cross. A monohybrid cross is a cross between two parents that focuses on one trait instead of multiple, hence the prefix mono, which means one. These crosses help to determine what kind of phenotypic traits the offspring will inherit from their parents. When making a cross, it's important to know the genotypes of the parents first. Let's use the genotype found for the brown-eyed father and the blue-eyed mother again. Big B, and little b. Monohybrid crosses are usually drawn as a 2x2 table. The genotypes of each parent will be placed above the first row of the table and to the left of the first column, as shown here. Whether you put the father's genotype or the mother's genotype above the first row won't matter. You'll still end up with the same results. To cross them, each letter will be placed in each box according to its row and column. So in the first box of the table, the letter above has a capital B from the father, and to the left of the first box is a little b from the mother. Both letters will be placed in the first box, as shown here. The same will occur for the remaining boxes, as you take each letter from the top row and from the leftmost column. Always keep the capital letter first when crossing, so it's easier to read when quickly looking at the cross at a glance. With the cross completed, this gives the possible genotypes or phenotypes the offspring will have. According to this Punnett square, 2 out of the 4 offspring will be heterozygous, big B, little b, where there is a 50% chance that the offspring will have brown eyes. Another 2 out of the 4 offspring will be homozygous recessive, little b, or there is a 50% chance that the offspring will have blue eyes. This can also be expressed in a genotypic ratio. There is a 2-to-2 ratio of big B to little b. Simplified, that would be a 1-to-1 ratio. This is the same ratio that would apply phenotypically. These results are ultimately only a probability of what kind of offspring the parents will have. Not all possible genotypes or phenotypes will be used. For example, the parents could have all blue-eyed offspring and not a single brown-eyed offspring, even though there is a 50% chance that either blue or brown eyes would show. But it at least gives us a good idea of what the chances are of having that specific trait.